Welcome back to the video. In some cereals such as rice and wheat, pollen grains lose viability within 30 minutes of their release. And in some members like Rosaceae, Leguminous, and Solanaceae, they maintain their viability for a month. You may have heard of storing semen, sperm of many animals, including humans, for artificial inseminations. It is possible to store pollen grain at a large number of species for years at in liquid nitrogen, which is at a temperature of minus 196 degrees Celsius. Such stored pollen grains can be used as pollen banks similar to seed banks in crop breeding programs. The pistil, megasporangium, ovule, and embryo sac. The gynoecium represents the female reproductive part of the flower. The gynoecium may consist of a single pistil, which is also known as monocarpillary, or may have more than one pistil, which is known as multicarpillary. When there are more than one, the pistil may be fused together. If they are fused together, they are known as syncarpus, or may be free, which is known as apocarpus. Each pistil has three parts: the style, the stigma, and the ovary. The stigma serves as a landing platform for pollen grains. The style is elongated, slender part beneath the stigma. The basal bulged part of pistil is the ovary. Inside the ovary is the ovarian cavity or locule. The placenta is located inside the ovarian cavity. Recall the definition and types of plantation you have studied in class 11th. Arising from the placenta are the megasporangia, commonly called ovules. The number of ovules in an ovary may be one, which is in wheat. The ovary may be one, which is in wheat, paddy, mango. To many ovaries in papaya, watermelon, and orchids. The megasporangium, which is known as ovule. Let us familiarize ourselves with the structure of a typical angiosperm ovule. The ovule is a small structure attached to the placenta by the means of a stalk called funicles. The body of the ovule fuses with funicles in a region called hilum. Thus, hilum represents the junctions between the ovule and the funicles. Hilum, it is an important line, junction between the ovule and the funicles. Each ovule has one or two protective envelope, which is known as integuments. Integuments encircle the nucellus, except at the tip. At the tip, where a small opening called micropyle is organized. Opposed to the micropylar end is the chalaza end, representing the vessel of the ovule. Enclosed within the integument, a mass of cells called nucellus. Cells of the nucellus have abundant food reserves. Located in the nucellus is the embryo sac of located in the nucellus is the embryo sac of female gametophyte. An ovule generally has a single embryo sac formed from a megaspore. An ovule has a generally single embryo sac formed from megaspore. Megasporangiasis. The process of formation of megaspore from the megaspore mother cell is known as megasporangiasis. Ovules generally differentiate a single megaspore mother cell in the micropylar region. This is micropylar end, this is nucellus, and this is megaspore mother cell. Megaspore mother cell. It is micropylar end, it is nucellus, it is two cells, one cell, and this is two cells, second cell. Again, this cell is divided into tetrad, megaspore tetrad, and this is known as micropylar end, where two synergids form and one egg is formed. This is known as central cell. This region is known as central cell, where two nuclei are present, and these three are antipodals. Now they form a very good structures in the form of these types. Now, it is the large containing dense cytoplasm and a prominent nucleus. The MMC megaspore mother cell undergo meiotic division. What is the most important of the megaspore mother cell under meiosis? Meiosis results in the production of four megaspores. Meiosis results in the formation of four megaspores. Female gametophyte. In majority of flowering plants, one of the megaspore is functional, while other three degenerate. Only the functional megaspore develops into female gametophyte, embryo sac. Only the functional megaspore develop into the female gametophyte, which is known as embryo sac. This method of embryo sac formation from the single megaspore is termed as monosporic development. Single megaspore is termed as monosporic development. What will be the polarity of the cell of the nucleus MMC, the functional megaspore and the female? All will have uh, nucleus will have 2n, and the functional megaspore will have n, and the female gametophyte will have n. Let us study the formation of embryo sac in a little more detail. Figure 2.8b. The nucleus is the functional megaspore divided mitotically to form the nucleus of functional megaspore divide mitotically to form two nuclei, which move to the opposite pole, forming the two nucleate embryo sac. Two more sequential mitotic nucleate divisions result in the formation of four nucleate and the later eight nucleate stage. Four nucleate and the later is eight nucleate stage of the embryo sac. It is of our interest to note that these mitotic divisions are strictly free nuclear. That is, nuclear divisions are not followed immediately by the formation of cell wall. After the eight nucleate stages, the cell wall laid down, leading to the organization of typical female gametophyte or embryo sac. Observe the distribution of cell inside the embryo sac, figure 2.8 BC. 6 of the 8 nuclei are surrounded by cell walls and organized into cell. The remaining two nuclei, called the polar nuclei, are situated below the egg apparatus in the large central cell. There is a characteristic distribution of the cell within the embryo sac. The three cells are grouped together at the micropylar end and constitute the egg apparatus. The three cells of group together at the micropylar end and constitute the egg apparatus. The egg apparatus in turn consists of two synergids and one egg cell. The synergids have a special cellular thickening at the micropylar end known as filiform apparatus. Spatial cellular thickening, which is also known as filiform apparatus, which play an important role in the guiding of the pollen tube in the synergid. Three cells are at the chelazal end are known as antipodals. The large central cell, as mentioned earlier, has two polar nuclei. Thus, a typical angiosperm embryo sac at maturity have 8 nucleus and 7 cells. It is very, 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 very important. Pollination. Pollination. In the preceding section, you have learned that the male and the female gametes in flowering plants are produced in the pollen grain and the embryo sac, respectively. Male gametophytes are produced inside pollen grain. Pollen grain are the not male gametophyte. They are produced inside pollen grain. It is one of the most toughest. Generally, students thinks that pollen grain is the male gametophyte, but it's not like that. Male gametophytes forms inside pollen grain, respectively. As both types of gametes are non-motile, they need have to be brought together for fertilization to occur. How is this achieved? Pollination is the mechanism to achieve this objective. Transfer of pollen grain seed from the anther to a stigma of pistil is termed pollination. Flowering plants have evolved an amazing array adaptation to achieve pollination. They make use of external agents to achieve pollination. Can you list the possible external agents? Kinds of pollination. Depending upon the source of pollen, pollinations can be divided into three types. Autogamy. In this type, pollination is achieved within the same flower. Transfer of pollen grains from the anther to the stigma of the same flower. In the normal flower, beach opens and exposes anther and the stigma. Complete autogamy is rare. Autogamy in such flowers requires synchromy in pollen release and the stigma receptivity, and also the anther and the stigma should lie close to each other so that self-pollination can occur. Some plants such as viola, which is also known as common pansy, oxalis, and commanila, produce two types of flowers. One is chesmogamous flowers which are similar to flower of other species with exposed anther and stigma, and clistogamous flowers which do not open at all. This means that clistogamous is closed, means anther and stigma are closed. In such flowers, anther and stigma lies close to each other. When anther desiccates in the flower buds, pollen grain comes in contact with the stigma to affect pollination. Thus, clistogamous flowers are invariably autogamous as there is no chance of cross pollen landing on the stigma. Clistogamous flowers produce a seed set even in the absence of pollinators. This is also an most important line. This is more often can be asked in exams. Do you think that clistogamous is advantage or disadvantage to the plant? No, it may be the advantage and disadvantage both for the plant. Geitonogamy. Transfer of pollen grains from anther to a stigma of another flower of the same plant. Means there is same plant, but the flower is different. Although geitonogamy is functionally cross-pollination involving a pollination agent, genetically they are similar. Genetically it is similar. Autogamy, since the pollen grain come from the same plant. This is also a type of autogamy because the seeds and the comes from the same plant. Xenogamy. Transfer of pollen grains from anther to a stigma of a different plant. This type of pollination which during pollination brings genetically different types of pollen grain to the stigma. Agents of pollination. Plant uses two, biotic and abiotic. In biotic, winds and water, and abiotics, animals. Agents to achieve pollination. Majority of plants uses biotic agents for pollination. Only a small proportion of plant uses abiotic agents. Pollen grains coming in contact with the stigma is a chance factor in both wind and pollen. Pollen grain coming in contact with stigma is a chance factor both in wind and water pollination. To compensate for these uncertainties and associated loss of pollen grains, the flower produces enormous amount of pollen when compared to the number of ovules available for pollination. Pollination by wind is more common amongst abiotic pollination. Wind pollination also requires the pollen grains are light and non-sticky so that they can be transported in wind currents. They often possess well-exposed stamens so that pollen are easily dispersed into the wind currents, and large, often leathery stigma to easily trap airborne pollen grains. Pollen Wind pollinated flowers often have a single ovule. In each ovule have a numerous flower packed into an inflorescence. A familiar example is the corn. Corn is an example of wind pollination. Corn cob. The tassels means a stigma. A stigma. You see are nothing but the stigma and the style which wave in the air to trap pollen grains. Wind pollination is quite common in grasses. Pollination by water is quite rare in flowering plant and is limited to about 30 genera. Pollination with water is only limited up to 30 genera, mostly monocotyledons. As against this, you would recall that water is a regular mode of transfer for the male gamete among the lower group plants, such as algae, bryophytes, and pteridophytes. Uses water as transporters for its pollens. It is believed, particularly for some bryophytes and pteridophytes, that the distribution is limited because of the need of bryophytes and pteridophytes are limited because of the need for the water for the transport of male gamete for fertilization. Some examples of water pollinated plants are Vallisneria and Hydrilla, which are grown in freshwater, and several marine seagrasses such as Joystra. Vallisneria, Hydrilla, and Joystra. Not all aquatic plants use water for pollination. In a majority of aquatic plants such as water hyacinth and water lily, the flowers emerge above the level of the water and are pollinated by insects or wind, as in most of the land plants. In Vallisneria, the female flower reaches the surface of water by the long stalk, and the male flowers or pollen grains are released onto the surface of the water. They are carried passively by water currents. Figure 2.11a. Some of them eventually reach the female flowers and the stigma. In another group of plants, water pollinated, such as sea grasses, female flower remains submerged in water and the pollen grains are released inside the water. Some of them reach stigma and achieve pollination. In most water pollinated species, pollen grains are protected from getting wet by a mucilage covering. Both wind and water pollinated flowers are not very colorful and do not produce nectar. What would be the reason for this? Because they do not need uh, flies and birds for its pollination, so they not require color. Majority of flowering plants use a rare a range of animals as pollinating agents. Bees, butterflies, flies, beetles, wasps, ants, moths, birds, beaches, sunbirds, and the hummingbirds and birds are the common pollinating agents. Among the animals, insects, particularly bees, are the dominant biotic pollinating agent. Even larger animals such as some coyotes, lemurs, arboreals, tree-dwelling rodents are often even lizards, uh, gecko lizards, garden lizards have been reported as pollinator in some species. Often flowers of animals pollinated plants are specifically adapted to a particular species of animals. Majority of insect pollinated flowers are range, colorful, fragrant, and rich in nectar. When the flowers are small, a number of flowers are clustered in an inflorescence to make them conspicuous. Animals are attracted to flowers by colors and fragrance. The flower pollinated by flies and the beetles secretes fall odor to attract these animals. To sustain animal visits, flower has to provide rewards to the animals. Nectar and pollen grains are the usual floral rewards. For harvesting the rewards from the flower, the animal visitor comes in contact with the anther and the stigma. The body of the animals gets coated with the pollen grains, which are generally sticky in animals pollinating flowers. When the animal carrying pollen on its body comes in contact with the stigma, it brings out pollination. In some species, floral rewards are in providing safe places to lay eggs. For an example is that the tallest flower, Armophilus, the flowers is itself are 6 feet in height. A similar relationship exists between species of a moth and the yucca plant, where both species moth and the plant cannot complete their life cycle without each other. The moth deposits its egg in the locule of the ovary and flower turns get pollinated by the moth. The larva and the moth come out of the egg as they start developing. Why don't you observe some flower of the flowering plants or any others available to you? Cucumber, mango, peepal, coriander, papaya, onion, lobia, cotton, tobacco, rose, lemon, eucalyptus, banana. Try to find out which animals visit them and whether they could be pollinators. You will have to be patiently observe the flowers over a few different times of the day. You could also try to see whether there is any correlation in the characteristics of a flower to the animals. What carefully observe the visitor come in contact with the anther and the stigma, as only such visitors can bring about pollination. Many insects may consume pollen and the nectar without bringing about pollination. Such floral visitors are referred as pollen and nectar rovers. You may or may not be able to identify the pollinator, but you will surely enjoy your efforts. For today, up to there. If you like the session, like it, and if you have any comment, please drop your comment below.